So yeah, that intro is a little bit late. I should have introduced myself this morning when I was opening up that session. So, but better late than never. So yeah, so what I'm going to do is talk for a few minutes about patient susceptibilities in drug safety assessment. And I mean, I've worked in pharma. I've worked in pharma for a long time. I'm a pathologist by training. I worked in tox pathology for a long time. And fundamentally, I believe that we do a pretty good job in drug safety assessment. But that said, I also think that there's really good room for improvement. And so I'm going to try to be balanced in how I present this. But I think most of you are probably aware that we use a lot of different modeling platforms in our typical approach to drug development, not just safety assessment. And if you look across the sort of biological complexity and even in vivo relevance of those platforms, the animal systems truly are apical. And they're not only apical because of their biological complexity, but actually where we put them in the overall development scheme. So we use them for lots of different things, even beginning back at the target ID, target validation. But they are strategically positioned very late in development, and in particular enabling some very critical decisions right before we go into clinical development. So we're highly dependent on those systems for making some very, very important decisions. And I think most of you recognize that in the safety space that the studies that we do are fairly standardized and regimented and intended to be fairly holistic in their ability to identify safety liabilities that we need to be concerned about as we move into patients. So the animal systems in this slide has a cardiovascular bias to it, obviously, because mostly that's my area of interest and dare I say, slight expertise, but it's illustrative, right? So we depend on those animal systems because we have a fundamental belief in the conservation of biology, response to injury, pathobiology, pharmacology, and all of those things. And I think that there's a fair rationale for that dependence. The cardiovascular system is a perfect example. Very similar anatomy, very similar physiology. Actually, absolutely no doubt that there are differences in physiology in particular, and those things have to be accounted for. But overall, there's more conservation than not in that system, which is why we're heavily dependent on animals as a surrogate for the human system. So in the past, and this is one of those studies or assessments that's often shared, to rationalize the effectiveness of our typical approach to safety assessment. And this was a study that was done back in 2000 by a HESI working group. And in this particular case, the data has a bit of a bias in that they looked at clinical outcomes and then ultimately looked retrospectively at the concordance of the animal studies. They looked at phase 1, so you're typically looking at fairly acute toxicities, liabilities associated with those drugs. And as they looked across organ systems, in general, there's pretty good concordance. And there's better concordance in some places than others. So I say that to say that in this particular case, where again it's biased towards positive predictive value because they are looking for that concordance, there oftentimes are direct correlates between what happens in animals and what happens in patients, and more often than not, much more often than not, thank goodness, we protect patients from untoward harm in our early clinical trials. That said, there are bad things that happen, very rarely, fortunately. You all are probably familiar with the most recent occurrence, which was the SPIAL trial in early 2016, late 2015, where there were patients who had an unfortunate reaction to a compound and actually caused some irreversible harm. And in fact, there was a patient who died in that study. The previous most notable occasion of this was 10 years prior to that, and it was the Tegenero trial in the UK, and it was associated with monoclonal antibody therapy, and there were 5 or 6 patients who developed a cytokine storm reaction to that compound, ended up in intensive care, and unfortunately none of those patients died. And in these particular cases, there were not, at least in the prospective assessments, evidence that these liabilities were going to occur. Each and every time this happens, there's a significant sort of retrospective evaluation, and there's some change to the way that we approach these things to make sure that these things don't happen again. But the reality of it is as good as the job as we do, there are bad things that happen, and we make every attempt to try to mitigate these kinds of occurrences. So in those two cases, those were both acute toxicities, right? They were phase 1 clinical trials. You know, the most unfortunate part of that are these normal, healthy volunteers typically that these things are happening in, which just makes that worse. But a class or a group or a spectrum of potential drug-related issues occurs in a very different context. And so this is an example of Bardock Sloane is an NRF2 activator, and it's still actually in the clinics, being evaluated for a number of diseases, particularly chronic progressive diseases that have some oxidative stress component to them, and that's what this drug is intended to modulate. But in this particular trial, and there had been previous phase 1, phase 2 trials in patients that didn't have a severe renal disease, didn't last as long as this particular trial did. But as the trial progressed, in fact, one, it didn't have any effect on their primary outcome of interest, which was the occurrence or onset of end-stage renal disease. But worse than that, it actually increased incidence of cardiovascular-related deaths in patients who had or were hospitalized with heart failure. So these are the troubling kinds of things that we deal with, where you have a compound in a very complex context in patients who are very, very ill, and you can't detect these changes until you've got large numbers of patients who ultimately have been exposed, looking at them over long periods of time. So this is a completely different challenge than those phase 1 acute toxicity kinds of things that oftentimes gets the most interest. If you look at, everybody in here knows that the pharmaceutical industry is really challenged with a high rate of attrition in our business. Most of what we do fails. You could question the wisdom of somebody deciding to do that kind of thing for a living. You just have to get comfortable with that idea, but I do think we make incremental progress, and when we get it right, we get it really right, and patients significantly benefit. But the reality of it is that we do have a lot of failures, and if you look in particular at the safety-related failures, most of our safety-related failures are happening before we actually even get into patients, because we detect liabilities that are not only development limiting, but they're not things that we want to expose patients to, either because of the severity of the liability or where the liability is occurring relative to where we think we need to be to get efficacy in patients. So the bottom line is that we protect patients, for the most part, by getting rid of stuff before it ever gets to the patient. Now, the downside to that is it's very difficult to understand concordance when you never actually took it in and gave this presumptive liability an opportunity to play out in patients. But that's the better way to go. It's the more conservative approach, and I think it's the right approach. That said, there are also toxicities that we don't detect until we get into the clinical setting, and again, the most troubling ones are the ones that are occurring late in development. So this is a significant challenge. These are more chronic toxicities. They're toxicities that are playing out in patients, and so there's some synergy with either the patient's disease or their genetic susceptibility and those kinds of things, which makes them very difficult to understand. That said, and this was actually brought out yesterday when we were talking about reproducibility, it's not hard to find reasons for why there would be disconnects between what happens in some of our animal studies and what actually happens in patients, when you consider the fact that we're generally looking at young, healthy animals that are very genetically homogeneous and patients that are essentially not that way. We also have very controlled environments, and we treat patients that are not that way. A lot of our drug development is for chronic progressive diseases they're very messy, they're very chronic, and they're a very challenging biological venue in which to work. The reason why, a significant reason why our models are as controlled as they are is that in the pharmaceutical industry, we're in a sort of in the decision-making business, right? We've got to have data that enables to make a decision about whether we progress a compound or not, and if we're going to progress it, what the risks are and how we're going to manage those risks. As a consequence of that, our modeling platforms are built, constructed to enable decisions. And so the more discrete the data, the more discrete the information you get from that model, the easier it is to make a decision. Now, the downside to that is that the more controlled that platform, the less patient-like it looks. And so there's a bit of a dichotomy there. In addition to the environmental and the biological issues, oftentimes in our studies we're measuring different things, right? So one of the upshots to doing animal studies that keeps pathologists employed is that we do fairly invasive and detailed assessments of those animals. That's good and bad, right? We're probably describing some morphologic things that would never be detected in a patient and may be completely inconsequential, but we're keying on them because we can see them. By the same token, there are different ways that you evaluate health in patients, and some of it is just them telling you how they feel, and we don't get that kind of information from animals. So even in our assessments, there are differences that might account for some of these disconnects. As I mentioned, a lot of the drug development that we do is for chronic progressive diseases, and it's a very challenging drug development setting. And this is a table pulled out of an old diabetes trial, 2009. But just to give you a sense of how complicated these patients are, right? This is not atypical for a patient, an older patient in a Western country. With high rates of cardiovascular disease, high rates of renal disease, metabolic disease. Again, a really, really messy biological, pathobiological context where we're trying to extrapolate what we learn from a completely different biological setting. That said, our animal studies are not squeaky clean. They also have comorbidities, and there are things that we have to ultimately read around occasionally. So, for example, the rodents, those of us who look at rodent tissues every day are used to recognizing a progressive cardiomyopathy that actually worsens as the animals get older. There is also, too, a chronic progressive renal disease that also becomes more severe as those animals get older, and then some pre-existing vascular disease that occurs both in rodents and in dogs, and also some valvulopathies. Well, all of those changes could be drug-induced changes. Those processes could be exacerbated by drug. And so we're constantly having to make decisions about whether we think some change in what we expect to see in one of these conditions is truly a drug-related event and relevant to a patient. So, much cleaner background than certainly our patients, but not a completely clean background. And so there's the complexity of having to read around these things, but also, too, they represent susceptibilities in those animal models, and that's part of that modeling system that we have to deal with. One of the therapeutic areas where we as a society typically tolerate more toxicity than others is in cancer drugs, right? Because of the severity of that disease, there are drug-related liabilities that are taken forward and managed when there is the opportunity to potentially significantly extend a patient's life. And so it's a place where we are used to seeing toxicity, for one, and have an opportunity to understand concordance between the animal models and in patients. And I think one of the things that we don't appreciate well enough, and I think these tables, which is basically a spectrum of cardiovascular liabilities associated with a whole portfolio of cancer drugs, and that is the relatively low incidence of most of those liabilities. So those are individual patient kinds of things that may be susceptibilities represented by the stage of their disease or some genetic susceptibility, those kinds of things. So the last thing in one of the cancer drug-related cardiotoxicity is one of those areas that I've been involved with in trying to partner with clinicians. And it's interesting the difference in sort of the perspective of a cardiologist and an oncologist, right? The oncologist wants to do nothing that's ultimately going to hamper their ability to significantly manage that cancer. The cardiologist is obviously sensitized to, well, how do we do that but minimize the liabilities associated with the cardiovascular effects? And so this is a challenge even for the folks clinically. One, to be able to recognize that these things are happening because there's a small proportion of patients, and how do you ultimately not undermine the benefit that the patient's going to get and still manage these challenges. from my perspective, the best opportunity we have is to know that these liabilities are out there and have a better sense of what patients are most susceptible and be able to be proactive about it. So those are the challenges. And again, as I mentioned earlier, a smart person just wouldn't do this for a living, but there are things, I think, and ways for us to get better at what we do. And there's a number of potential approaches that I'm going to talk through just briefly. So this is a more recent analysis that was done by the IQ Consortium, which is a pharma consortium group, people working on things of common interest across companies. And one of the significant interests was actually building on that Olson analysis that I showed you. So we're updating it, and actually they've taken a bit of a different tact in that these are compounds that were terminated, and I think there was like 11 or 15, somewhere between 11 and 16 companies that have contributed to this. So it's a fair representation of the portfolio of a number of pharmaceutical companies. And it's much more prospective. So these aren't things that specifically had outcomes in patients and they're tracing back the concordance. So this is much more balanced between the ability to assess positive and negative predictive value. So the positive predictive value is still undermined by recognizing that for significant liabilities, we just don't take those drugs into patients, right? So your prevalence rates are pretty low, and that undermines your positive predictive value because you got rid of them before you went into a patient. But what is significant here, and I think what they've emphasized in this paper, and I think rightly so, is that our current approach to safety assessment has a very high negative predictive value, meaning if you've got a lack of toxicity in your animal models, and this distinguishes across species, and there's a fair concordance across those species, that you can be fairly confident that you're going to be safe in patients. And so there's a lot of confidence that comes from those kinds of assessments. And so this is part of the balance, right? So it's easy enough for us to pick out places where things go awry and pick on the deficiencies or the gaps in the system, but overall, our current approach does a pretty good job of protecting patients. That said, again, I think there are opportunities to be better. And so one of the things that I'm interested in, particularly as I've gotten in animal modeling, not just in safety assessment, but more broadly than that, one of the things that has struck me is that if I'm a patient and I go into my physician, regardless of why I'm there, ingrown toenail or angina, there's a basic set of physiologic parameters they're always going to measure, right? So we capture this package of basic physiologic data whenever you walk into the office. And a lot of that stuff doesn't routinely get measured in our animal studies. There are good reasons for that because it's not efficient to do that. We don't have the technology to do that. The data is noisy because you take the animal out of the cage and try to take his heart rate, or you got to anesthetize, blah, blah, blah. So lots of reasons why that occurs. But not unlike the microphysiological systems that we just talked about, there's technology that is developing that's going to give us a better opportunity to monitor our animals more holistically from a physiologic and even a behavioral perspective that I think is going to offer us great context for the things that we typically measure: histopathology, clinical chemistry, gross findings, those kinds of things. So I think there's really good opportunity to monitor our animals in a much more holistic way. So I think that's an opportunity, and we'll see things happen that we wouldn't in our normal way of measuring things. The other thing is that if you put some of these compounds into in diseased models or stressed models, actually you see different things, right? So you've got a couple of choices. One, you can try to replicate a disease context in which you're moving into in your patient, right? So to provide that sort of connection and understand the susceptibilities that might be coming with that. Or you can take a system, and the cardiovascular system, again, is where I spend most of my time, and recognize that there's a fair bit of reserve in the cardiovascular system. There is the potential for drugs to have some bioactivity in that system for which the cardiovascular system compensates, and so I don't see it as pathology or toxicity. But in a patient who doesn't have that reserve, that could be a completely different picture. Well, I can replicate some of that stress and loss of reserve in animal modeling systems, and in fact, folks have published some of that kind of work. And here's just another example. Gene Herman did a lot of work when he was at FDA looking at doxorubicin and susceptibilities, and in this particular case, he looked in hypertensive rats. Well, just so happens that hypertensive rats, particularly if you're interested in going into hypertensive patients, that is a model that's got a fairly good history of replicating or translating from animals into man. So there are, again, opportunities to look at specific susceptibilities and actually model those in animal systems. As we talked a little bit about yesterday, this isn't entirely a technical issue, right? So there are cultural and behavioral components to all of this. And as I mentioned before about being in a decision-making business and trying to get very discrete data so you can make decisions, there's been some reticence in creating opportunities for equivocal data or grayness in the information that we get. But that's changing. And in particular, this is a paper that was published by a working group in the Society of Toxpath, recognizing that in fact that there are probably some animal models of disease or stress that are relevant for specific applications. Nobody's making any proposals about moving into this as a routine, but certainly opportunities for us to push our systems in human physiologically relevant ways and try to understand potential susceptibilities. Won't talk about this because Paul, I think, Paul and Megan did a really good job of talking about iPSC, patient-derived iPSCs and the ability to replicate some of these susceptibilities in vitro, and I think there's a lot of really good mechanistic opportunities there. The other thing that I'm working on with actually some folks at the FDA is actually looking at much more mechanistic approaches, right? So this idea that our current approach is very phenotypically anchored, and we talked a little bit about this yesterday. If we truly understood the mechanistic bioactivity of our drugs, could you start to extrapolate or reflect that into what we know about human susceptibilities and get a better sense of when there's real liabilities to be had there? So I think it's another approach very early in our thought process about doing this. So in summary, because I've got my red light blinking and so I have to lead by example here, which I've already screwed up, but I think our current approaches in preclinical safety assessment it largely protect patients from certainly irreversible injury, but there are certainly examples of where that's not the case. I think the evolution of precision medicine is going to change expectations, about even in the safety world, around our ability to understand specific challenges with specific patients. I think the advances in technical capabilities is going to enable that. So I think that that's a good thing, and we've got to figure out how to ultimately manage that. So, as I mentioned earlier, I think a big part of this, I mentioned it in the introduction this morning, a big part of this is understanding what susceptibility is in the patient. So that to me is one of the things I'm excited about in precision medicine, is actually getting better characterizations of patients. So we have a better understanding of what it is ultimately that we're trying to model. So thank you for that.